antibiotic belong to 5 groups like cell wall inhibitor, cell membrane inhibitor, metabolism inhibitor, DNA gyrase inhibitor, and protein synthesis inhibitor. These can be cidal or static. Cidal, which kill one microorganism. So beta lactamases, they kill the microorganism. Vancomycin, fluoroquinolone, and aminoglycoside. So protein synthesis inhibitor are macrolide, chloramphenicol, tetracycline, and linezolid. These are static. They decrease the growth of bacteria so that body can kill them in the meantime. Cell wall inhibitor, we have beta lactam, like penicillin, cephalosporin, carbapenem, and monobactam. Vancomycin. Third is phosphomycin, cycloserine, and bacitracin. So how they are acting? So first, inside the bacteria, NAG is converted into NAM, which is inhibited by phosphomycin. Peptide chain is also produced inside bacteria, which is inhibited by cycloserine. Then these NAG, NAM units and peptides are exported to form the cell wall, and which is inhibited by bacitracin. Then these NAG, NAM, and peptide chains are cross-linked, and transpeptidase cross-link the these peptide molecules. This is inhibited by beta lactam. Transglycosylation and transpeptidation. These peptide chains and glucans like NAG and NAM are cross-linked, and which is inhibited by vancomycin, which bind to D-ELA. So the D-ELA and D-ELA, this area, D-ELA, vancomycin bind here and inhibit the cell wall synthesis. If there is no cell wall, then membrane permeability to the water will increase, and bacteria will die because of bursting from extra water inside. Mycoplasma has no cell wall, so these group are not active against mycoplasma. So how can we remember these 5 groups? So mnemonic is bacterial cell wall binding figures. Beta lactam, cycloserine, and W for vancomycin, B means bacitracin, and F for phosphomycin. Cycloserine, if I say psycho, so psychosis may, this drug is used in TB also, but the side effect is psychosis. Bacitracin is topical. If the staff healthcare workers have found to be Staph aureus MRSA in the nasal area, we can use the topical ointment of bacitracin or mupirocin to kill those Staph aureus, and this is therefore used for the prophylaxis of MRSA. Phosphomycin, 1 pouch is used and given to the patient in case of UTI. So gram-positive bacteria can be cocci or bacilli. Cocci are staph and streptococcus. Bacilli are bacillus, B, C, D, and L. So bacillus, clostridium, diphtheria, and listeria. Bacillus, diphtheria, and listeria are aerobic, and clostridium is anaerobic. Gram negative is cocci, Neisseria, and bacilli is sheep club, Salmonella, Shigella, H. influenza, Helicobacter, E. coli, Pseudomonas, and Klebsiella. So if any antibiotic do not act against the sheep club, then it is called narrow, and if the antibiotic kill all these bacteria also, this is wide spectrum antibiotic. Now we study beta lactam group. Beta lactam group has a beta chain and a second side chain. So it contains penicillin, cephalosporin, carbapenem, and monobactam. Monobactam only has one beta lactam ring. There is no second ring. So in penicillin we have penicillin G. Penicillin G has 5 shortcomings. It is short T half because it is tubular secreted from the tubules. It is secreted out, and orally it is acid labile, so it is not orally active. So we have to give injection, and in short duration if we have to give injection, then it is not a very good drug, and it has a narrow spectrum, so it is not active against those sheep club groups, and resistance is a problem in this, and allergy is also there. So to overcome short T half, we what we can do is tubular secretion competition. We can competing drug we can use, that is probenecid, or we can use a depot drug, means we in the IM intramuscles we attach a benzathine group or procaine group to release the drug slowly into the blood, so that if 100 molecules go, they are tubular secreted in 2-3 hours. Then other due to concentration gradient, muscles act as a reservoir of the drug and keep on giving 100 molecules, so the action become longer, and benzathilin is the longest acting penicillin. Oral, if these were not oral, to overcome that we have vodka and vodka penicillin include penicillin V, phenoxymethyl penicillin, oxacillin, cloxacillin, dicloxacillin, amoxy and ampicillin. These are acid resistant penicillins. Then extended spectrum penicillin. Extended spectrum penicillin mnemonic is ACT map. So these are active against Salmonella, Shigella, E. coli. A for MP, amoxy. C for carbonicillin. TY for ticarcillin. M for meslo. A for azlo. P for piperacillin. So CT map is active against Pseudomonas, and MAP is active against Klebsiella also. So MAP is active against Salmonella, Shigella, E. coli, Pseudomonas and Klebsiella. CT map is against Salmonella, Shigella, E. coli and Pseudomonas, and ACT map against Salmonella, Shigella and E. coli. This is the extended spectrum penicillin. Now resistance. Resistance is first the bacteria get a gun, so they kill the antibiotic by hydrolyzing it, so they have beta lactamases or penicillinases. So to overcome that penicillinase, the antibiotic we give bulletproof jackets to the antibiotic, that is penicillinase inhibitor. So penicillinase inhibitor themselves do not work, we have to give antibiotic with them, and so clavulinate, sulbactam and tazobactam are the penicillinase inhibitor or beta lactamase inhibitor. We give amoxiclav, MP sulbactam, pipratazobact or ceftazidime, avibactam, newer group, ceftazidime and avibactam, very good antibiotic we give. But then the bacteria develop change the penicillin binding protein, where penicillin was binding and inhibiting transpeptidase. The bacteria modified that, so PBP2A is formed, so this leads to MRSA formation. So methicillin resistant Staph aureus group is formed. So if a bacteria is MRSA positive, so before methicillin sensitive Staph aureus is there, so beta lactamases resistant penicillin are there. So condom group like oxacillin, cloxacillin, oxacillin, neficillin and methicillin are gun-proof penicillin. So if bacteria has gun, either we can have a gun-proof jacket with the penicillin, or penicillin themselves is gun-proof, like a Rajnikant penicillin. The beta lactamases have no effect on them. So these are condom or monk penicillin. These are like methicillin, oxacillin, neficillin and cloxaoxacillin. If methicillin resistance come, then these all antibiotics are not of any use. So MRSA comes because SCC has chromosomal MAC-A gene in the bacteria which produces PBP2A. MRSA is formed. So then we have to give vancomycin. Vancomycin bind the DLR group and it act by that. But if DLR is converted to D-lactate, that is cell wall precursor is altered, then we will have vancomycin resistant Staph aureus or vancomycin resistant Enterococcus. So in between we have one species like visa. Visa is extra cell, these are cell wall peptides, so they will have extra DLA here. So in between the antibiotic will bind to that and cannot act. If we give more antibiotic, then these antibiotic will be able to act on the bacteria. So visa, but at H visa is also there. In the report we will see that VSSA is there, but clinically when we give vancomycin, the patient is not getting better. So in that case we send into library that to the laboratory that H visa may be there. So there may be few colonies which are resistant to vancomycin. Then if lab says okay it is H visa, then we say it is resistant to vancomycin, then we go to daptomycin or teicoplanin in group of drugs. Allergy, cross allergy is there except in monobactam. Monobactam has a group called astrionam. Intradermal testing we do, which is not 100% proof. So we then after intradermal allergy testing we give slow IV infusion for 10-15 minutes and see if there is no allergy.